let's pause here to go over what I call bookkeeping to keep track of what the substrates and products are for each of these metabolic pathways. So starting with glycolysis, our first substrate is glucose, and from that we get 2 pyruvic acids, 2 ATP net, remember total we've made 4, and 2 NADHs. Now from here on out we're going to be keeping track of everything per glucose. So the pyruvic acids go into acetyl-CoA formation, and they came from glycolysis, and we make 2 acetyl-CoAs, 2 NADHs, and 2 CO2s. Now these 2 NADHs are going to go into the Krebs cycle, and from that we're going to get 4 CO2s, that's per turn. We have 2 acetyl-CoAs, each one has a turn through the Krebs cycle, so we get 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. Now these CO2s, the cell can no longer get any energy out of them, so they diffuse out. That's why you exhale CO2. That's why bacteria oftentimes will produce CO2. Also, we can measure their metabolic activity by how much CO2 they produce. These NADHs and the FADH2s are going to go to the electron transport chain, where we're going to be generating ATPs through oxidative phosphorylation. The 4 ATPs that we've made through glycolysis and the Krebs cycle have been through substrate level phosphorylation. So through oxidative phosphorylation, we can make 6 ATP per NADH and 2 ATP per FADH2. That's a pretty good exchange rate. So now we're going to talk about the electron transport chain, affectionately abbreviated as ETC here, and how we get these ATPs out of the NADH and the FADH2s. To begin with, we're going to go over a cartoon version of the electron transport chain. What happens is NADH donates a hydrogen to the first in a series of enzyme complexes that are going to pass that electron from enzyme complex to enzyme complex until it gets to the end of the chain. In the process, these enzyme complexes are going to be reduced. They're going to gain energy, and they're going to cycle back and forth as NADH and FADH2 bring in hydrogens. Now they're going to split these hydrogens. They're going to spit the protons, the hydrogen ions, to the other side of a membrane. And the electrons that came from the hydrogens are going to be passed down this chain, reducing these enzyme complexes so that they have the energy to pump hydrogen ions, to the other side of the membrane. So when you get to the end of the chain, we have built up an awful lot of hydrogens on the other side of the membrane, and they can't get back. We're going to be using that energy to make ATPs. Now this last electron is worn out. It doesn't have any more energy, so we have to get rid of it. So we have what's called a final electron acceptor that's going to grab that electron. In the process, it's going to grab some protons, further reducing the concentration on this side of the membrane. Now, before we go on to the more accurate representation of an electron transport chain, you'll notice that NADH starts out at the top of the chain, FADH2 starts further down. I like to use the slinky example. Say the electron transport chain is a set of stairs. Here the slinky is up at the top. It has more potential energy to go down the stairs. Therefore, it generates more ATPs, 3 of them. Now, if we start the slinky about 2 steps down, it doesn't have as much potential energy, and that's why we only get 2 ATPs out of it. Here's another way to remember it. FADH2, 2 ATPs, and NADH, you just have to remember it's 3. So, let's look at the more accurate representation. So here are our enzyme complexes. And so NADH here comes and donates a hydrogen to FMN, and it's going to be oxidized to NAD+, and it's going to go back to glycolysis, the Krebs cycle, and other pathways. FADH2 donates its hydrogens and the electrons with them to ubiquitone, which is second in the chain, so that's why we get fewer ATPs out of it. So FADH also goes back to pick up more hydrogen. The electrons that are donated are removed from the hydrogens, and the protons, the hydrogen ions, are pumped to the other side of the membrane. And you'll notice as each of these enzyme complexes are reduced, they use the energy from that reduction to pump hydrogen ions to the other side of a membrane. The electrons are tired, they're worn out, and we need to get rid of them. Otherwise, these guys can't go to the next enzyme complex, and then these guys have to wait here, and it just binds up the whole chain. So, anaerobic respiration, of which you engage in, and most microbes that we're going to be using in the lab are also aerobic. We take oxygen, and it is the terminal electron acceptor. Oxygen goes up here, grabs these electrons, gains a negative charge, picks up some of the protons that are hanging out here, and we get water. So that's why you have to breathe in. That's why we have to have, when we grow microbes, most of the microbes in the lab, we need to have oxygen present so that we can keep this electron transport chain going. When you are cut off from oxygen, we can't continue to pass electrons down the chain, and this whole system shuts down. And the cell has to go with just glycolysis. All of these protons up here, we call a proton motive force. We also call it chemiosmosis. We have osmotic pressure building up here. The hydrogens want to get to the other side through diffusion, but they can't get past the negatively charged phosphate heads of the membrane. So what we do is we have an enzyme called ATP synthase that is going to take advantage of this proton motive force of chemiosmosis to make ATP through oxidative phosphorylation. ATP synthase has a port, a hole, that allows the hydrogen ions to diffuse through and come out into the area with fewer hydrogen ions. In the process, this part of the enzyme spins just like a turbine, and it takes ADP and inorganic phosphate and smooshes them together to make ATP. It almost looks like a turbine in a hydroelectric plant. So you can think of this as water behind the dam. The water on this side of the dam is higher. It wants to flow to the lower area through gravitation. We open up a hole, the rushing water causes the turbine to spin, and we create energy. In this case, it's ATP. In hydroelectric dams, it's electricity. That's how we use the energy from NADH and FADH2 to make ATPs, and that's why we need oxygen to do this. Now, if an organism that is aerobic is cut off from oxygen, it is limited to the ATP that it can make through substrate level phosphorylation through glycolysis, because if the electron transport chain shuts down, so does acetyl-CoA formation and the Krebs cycle. So let's talk about where all of this is happening. Let's start with what you're familiar with, in the mitochondria in your eukaryotic cells. This membrane here is the inner membrane or cristae of the mitochondria. Then we have the outer membrane. In this inner membrane space is where we're building up our proton motive force. They want to get into the matrix. This is the matrix. So we're building up a high concentration of hydrogen ions in the inner membrane space, and they're coming into the matrix through ATP synthase. Now we've got just about exactly the same setup in aerobic bacteria, but they don't have mitochondria. So what happens is, this is the cellular membrane, this is the periplasmic space, and this is the cytoplasm. So hydrogens are pumped to the space between the cytoplasmic membrane and the cell wall. They want to get into the cytoplasm. They come in through ATP synthase. So that's how it works with aerobic respiration. Well, what about anaerobic electron transport? Used to be that we thought that if you didn't use oxygen, you were limited to glycolysis and fermentation. So Pasteur called fermentation anaerobic respiration. Well, as it turns out, we have since found organisms that use electron transport chains, but they use a different terminal electron acceptor. So we no longer consider fermentation to be respiration because it doesn't involve an electron transport chain. Now, anaerobic electron transport is anytime you have this type of electron transport chain pumping hydrogens to the other side of a membrane and using an enzyme to create ATP through anaerobic respiration, anaerobic phosphorylation. The book has several examples, but my favorite one for an anaerobic electron acceptor is sulfur. Sulfur is close to oxygen on the periodic table. It likes to pick up electrons, so it comes and picks up electrons, makes it negative, picks up 2 hydrogen ions, and we get hydrogen sulfide, or rotten egg gas. So if you're in a swamp and you're seeing bubbles coming up from the mud where there's no oxygen, those are rotten egg gas bubbles. And that's why swamps generally stink, is because of the anaerobic respiration of these organisms that do not use oxygen as their terminal electron acceptor. Go and read the book. Make sure that you know what the other electron acceptors are. But this is my favorite example because it pairs with oxygen so well. So here is the reminder of what you need to get from this section. In general terms, describe the 3 stages of aerobic glucose catabolism, glycolysis, the Krebs cycle, and the electron transport chain. Oftentimes they include acetyl-CoA formation in the Krebs cycle. Including their substrates, products, and net energy production. Discuss the roles of acetyl-CoA, the Krebs cycle, and electron transport in carbohydrate catabolism. Contrast electron transport in aerobic and anaerobic respiration, we just did that, and be able to describe the function of ATP synthase and how it interacts with the electron transport chain.